everyone for being here. This is our second evening of having a Bobcat alumni from the CHBE department talk to our undergrads about their experiences as a practicing engineer. And tonight I'm really thrilled to have Ashley Schrammick join us. She is currently a process development scientist at GlaxoSmithKline in Hamilton. And when I was graduating with my bachelor's degree in chemical engineering, this absolutely would have been my dream job. I wanted to work in a company like that and somewhere in the mountains. So I'm thrilled to see that Ashley has found such a great position. She graduated from Montana State University in 2015 with degrees in chemical engineering and biological engineering. So she was a dual major. She did research with Dr. Ross Carlson and his lab group. And she also had summer internships in Hamilton while she was an undergraduate student. And so I'm really excited that she's going to be talking to all of you today about her experiences. It's only been 5 years since she graduated, so she has a lot of experience in the first 5 years right now, so to speak. And one little unknown fact about Ashley is in 2015, it was a very important year for Pi Day, because Pi Day happens every March 14th. And in 2015, it was 3.1415. And so there was a big celebration for Pi Day. And Tau Beta Pi, the National Engineering Honor Society, had a competition for the Pi Day t-shirt that year. And Ashley won that competition. So not only is she a talented engineer, she's also a very creative artist as well. So thank you so much, Ashley, for being here. And I'll turn the microphone over to you.
Sweet. Thank you for the intro. I kind of forgot about the Pi Day thing, so I'm glad you brought that up. I have a couple slides that I was going to just go through because, I don't know, pictures are probably more fun to look at than my face. So I'm going to try to share my screen, and I'll ask you, actually, host disabled attendee share screen. So maybe.
Give me a moment.
Okay, thank you.
Welcome. Now, while we're doing that, give me a moment. And
okay.
I am going to share the following screen. Students, you can scan that and register for the event while I change the settings on this. Okay, I'm having slight difficulty making this change, so if you can vamp for a minute, I will figure it out and get you on. Can you just give us an introduction to your role at GSK?
Sure, yeah.
Thank you very
much. Sounds good. So let's see. Yeah, I think I'm pretty much just going to talk about my experiences working in Montana post-graduation and using my degrees from MSU, and probably just touch on my current job, which is a process development scientist in GSK, and just general activities of what that means and what I do day-to-day activities. And yeah, I'm not good at vamping, I'm sorry.
No, that's fine. Dr. Carlson, I know that You're here for a special reason. Can you indicate what that is?
Special reason, other than to embarrass Ashley.
Ooh.
I mean, I guess it's good to see you again. Ashley Beck is back in Montana, and we're probably going to finally finish your paper, the one that you and Katie and
Oh, neat. Right
that'll
on.
hopefully happen here in the next couple months. It's been a long time coming, but we'll have 3 or 4 undergrads on the paper, and all of you will have a superscript saying current address, and you'll have something else listed.
You
Ooh.
should be good to go, Ashley. Sorry about the technical.
Okay. Oh, that's all right. I'll just do the whole screen since it's just PowerPoint. And cool. Can you all see that?
Yes.
Sweet. We'll get right into it. I'm going to see if I can get, or can you see my mouse when I like just kind of roll it over the PowerPoint slide or not so much?
Can.
Perfect. Okay, let's do it. All right, so I am going to start out by talking about Hamilton because that's where I grew up and where I currently am. So I was born over in Washington and grew up in the Bitterroot Valley, which is just south of Missoula if you're not familiar with the area. Hamilton has about 5,000 people who live in the town. And it's kind of unusual for Western Montana because it has 2 fairly major science facilities. So the first one is Rocky Mountain Laboratories, which is a government-run branch of the National Institute of Allergy and Infectious Disease, and that's also part of the NIH, so that houses one of the 13 BSL-4 labs in the United States. So it's kind of weird that that's in Montana. And it's definitely weird that it's in Hamilton, Montana. So we also have GSK Vaccines, which is where I currently work. The site was originally started by RML scientists, Edgar Reedby and John Cantrell back in the 1980s. It was bought up by Corixa in the 1990s and then finally bought by GSK in 2005. So we currently make adjuvants, which I'll talk about what the heck an adjuvant is in a couple minutes. But yeah, it's definitely a weird area, and you should check it out if you're interested in trying to stay in the state post-graduation. I'm familiar with the struggle of trying to find relevant work opportunities in Montana after graduation. So, you know, if you're interested in pharma or medical application, it's definitely an area to check out. I know there's a lot of great startups in the state, but it can definitely be hard to know where to look. So Hamilton's a great area to check out if you want to try to stay after graduation. So like Abby already said, I am a 2015 MSU graduate. I did the dual degree with chemical and biological engineering, so I ended up graduating in that December. Some other stuff I did while I was an undergrad, I was a RA on campus for 2 years. And then I had 3 different types of internships, which was interesting because, you know, one was with the government, one was with a startup, and then one was with an academic lab with Ross on campus. So I don't know if I have anything specific to say about that, but, you know, we can talk questions at the end if you want. So we'll talk about what I'm up to now. So I work at GSK Hamilton, which is a 35-acre campus, and we specialize in making adjuvants for vaccines. So adjuvants are used to make vaccines work more effectively, and they enhance the vaccine's ability to elicit a more strong, durable, and protective immune response. So we currently make monophospholipid A, or MPL, which is a bacterial-derived adjuvant. It's basically a detoxified version of a cell wall. So it makes, you know, when you got your little vaccine, it makes your immune system it's very highly visible to your immune system since it has that cell wall component. And we're not quite there yet, but we do have a new facility that is being constructed, and we will be making QS21. QS21 is a plant extract. It's a saponin that's actually from a Chilean tree. I'm going to totally butcher saying it, but it's Quillahaya saponaria. So that's the QS part of it, and then the 21 is the 21st peak in the chromatogram. So we broke ground, I think fall 2018. And if you can see my mouse, this is the new building up here. We'll get started on consistency lots this upcoming spring. So it'll be 1, 2, 3 years, and we'll be making product here soon, which is really exciting. Other stuff to point out, I guess, site-wise, this is our main facility right here for making the NPL, and the other buildings are like admin and QA, QC, other offices and stuff like that. So, oh, and then before I get too far, so we make the raw adjuvants at our site, but they get used as adjuvant systems downstream. So before they're actually combined in the vaccine, like there's AS01, so adjuvant system number 1 has MPL and QS21, and GSK owns 3 like proprietary blends of adjuvant systems that get used in vaccines. So yeah. So I just wanted to include this quick little slide as a visual to compare a vaccine with an adjuvant compared to a classical vaccine. So the adjuvant, gives a stronger initial response with your immune system, and then it provides a longer duration in protective immunity compared to a classical vaccine. And a real life example of this was fairly recent. GSK came out with Shingrix just a couple years ago. And compared to that, the previous vaccine was called Zostavax, and it reduced shingles by 51% in adults age 60 and up, but it only lasted about 3 years. So Shingrix with the adjuvant system has a much greater efficacy at 90%, and it's marketed towards 50 and up, but they're also trying to do more age groups as well. So just a quick example of how that gets applied. Cool. So what the heck do I do? I started as a contract worker actually, and I guess I want to bring that up as a a possibility for if you're looking for jobs post graduation. Contract work is a good way to get your foot in the door, right? But it's I'm not going to sugarcoat it. It's not the greatest. It has relatively lower pay and no benefits. So there's no health care or no vacation days. And you can't participate in company stuff like lunches or town halls and things like that because of some lawsuit that happened a couple of years ago with I think Microsoft, but don't quote me on that. Anywho, contract work is a great way to get started, and it actually worked out for me okay because now I have a full-time employee job. So as a contract worker, I was a quote-unquote associate scientist, and now I'm a quote-unquote process development scientist, but really I do the same exact thing I did during that contract gig. So happy it worked out, and pretty much get to do the same job, but pay and benefits, so that's great too. My job is process development scientist, like I said, and I work in MSAT. And MSAT is an acronym that stands for Manufacturing Sciences and Technology, and it's a model that's becoming more popular across the industry. So I don't know if you're Googling jobs or whatever, MSAT is definitely something that's popping up more and more. We only adopted it actually about 2 years ago after GSK bought a different company that I can't remember off the top of my head. And they used the MSAT model, and then we adopted it. So what is MSAT? It is a manufacturing support group. And the 3 main things that we work on are support for site manufacturing and general process stewardship. So that looks like, support for investigations, process impact assessments, inspections, process monitoring, education on site, tech transfers, all that jazz. The second thing we do is process science, and that is mostly focusing on downscale models of the manufacturing process and analytical test methods to support general process and method development. So that's, stuff like making process improvements or assay improvements, scale up technologies, looking at new options to implement into production, as well as troubleshooting and extended characterization of your product. So yeah, and the final thing is just validation. And that is a pretty big third thing that ends up being how we adopted ourselves to the MSAT model. We were just a development group that had process development and analytical development. And then we kind of just suckered in the validation group. So what they work on is really equipment and cleaning and automation and stuff like that. So typically I'm found in the lab or at my desk writing about the stuff I did in the lab. I'll occasionally have to go into production. And instead of reading the list off on the side there, I have some photos of what, just some examples of things I do day to day. So we're just gonna go and look at those. So troubleshooting and deviation investigations is a really big role, especially since currently we only make the one product and we're soon to make the other product. So if something goes wrong in production, it usually comes to me. So in the first two photos, there was a washer that was part of a diaphragm pump and somehow it broke. And basically that washer fell into our CIP detergent tank. So I was given a fresh washer and was told, okay, well, tell us how long that washer was basically in the tank so we'd know about when it happened. We could assess which lots it could have impacted and, all that good stuff with the deviation. So that was kind of a fun one just because I got to dissolve some things in the lab. The third photo was a Taxol leak in our lyophilizer. So that's a freeze drying apparatus, and this oil is for the vacuum pump. And since it was an aromatic hydrocarbon, we thought, okay, well, maybe we can shine a black light on it and see if there's any of that oil in the lyophilizer. So in the lab, I got to shine a little light on the sample and it totally worked, as you can see as the photo, but it didn't work on the coupon study that I did. So it was one of those things where the idea was there, but it didn't quite work out in execution, but it made for a good photo. So that's why I included it here. And also, you know what? Sometimes science doesn't always pan out. So that's why it's there. Right, and the third thing is particle investigations. Obviously, we don't want a particle in our product for patient safety and all that good stuff. This one was a fairly recent one where supply realized they had a particle. They thought it was probably parafilm that had entered the bottle, and it turns out it was. So just one of those things that ends up on my desk and look at under a microscope, which is, nice variety, I guess. All right, another thing I work on is scaling up or testing new technologies. So with the QS21 process, we're actually transferring the process over from a contract manufacturing organization or CMO located over in France. So we'll be making the product and they will still be making the product as well once we're up and running. We're trying to keep the process pretty much the same, but there's one part with the filtration step upstream that we wanted to improve. So basically, they have a version of their filtration process where they're working with this water-insoluble resin, and that gets caught into basically an overglorified tube sock, and then they have to clean the tank. Cleaning the tank is expensive, it's awkward. They made their own special tank and it had a whole host of problems. So we started looking at single-use systems. So that included testing a bunch of different technologies. So in the bottom photo here, this was just one of the options. It was a candle filter that we didn't end up working out, but it was kind of neat to play around with. But anyway, the whole point of this slide is to show like, okay, we tried it out on the small scale system, determined that, okay, yeah, we're going to capture this amount of the resin and it should work, scale up. Tried it again at the pilot scale. So this was just one filter with the rented out pilot version that can hold the filter. And then this was just a couple weeks ago in the new facility. We did a water run, and you know, things are still being built over there, which is why, hence the hard hats and all that good stuff. But just an example of what it is to look at scaling up and testing out new technologies. So the story is not done yet. Eventually we'll have about 8 of those and actual product running through, but that's to be coming in the spring. And then some of the stuff I do is a little bit random or one-offs, especially with deviations, but then they have more routine activities. So the top, this photo right here is an activity called bark screening. So since our QS21 is derived from tree bark, our organization provides support to a different CMO to ensure that the bark that is selected is going to have the right ratios that we're looking for in the saponin content. So since trees are natural products and there's going to be variation with the climate and just different trees have different profiles, so we have to screen the trees before we can select each lot. So I actually finished that up this week for the year, which is exciting for me. So that's more of a routine activity. Other things might include sample prep, which is what the second photo is more or less trying to say. The third photo is just, it's a microfluidizer. I provide support to Belgium often because they take our adjuvants and then make the adjuvant system. But they don't usually have the means to do like true process development. So I guess in an example in the coming weeks, I'll be using that type of machine to do a proven acceptable range study for them and provide data to them so that they can use it for an FDA observation. And then the bottom photo is just a little coupon study. These are what we call coupons. They're just slices of, in this case, stainless steel. And I think I was looking at if a resin could come off easily in our CIP system. So lots of kind of random things that I do. It's interesting and kind of fun, but yeah, I don't know. Do you guys have any questions? I can stop sharing my screen. That's all I had. So.
Thank you very much. So yeah, we have a question here from Reed that asks, you dual majored in bio and chemie. Did you have interest in pharma or take any pharma classes at MSU before you started work at GSK?
Okay, let me get my stuff figured out. Okay, cool. So there weren't, I wouldn't say there were pharma classes while I was there. We were just getting started on some of the biomedical classes. I was sort of interested in the pharma industry just because I liked the idea of, using my degrees for kind of a more healthcare related field. But yeah, that's, I didn't take any like additional classes outside of MSU.
Students, you can submit questions through the Q&A button on there on Zoom, or by sending a message to panelists and attendees. I have a question that came in via email today, which is, Are there a lot of PhDs at the campus at GSK in Hamilton?
That is a really good question. So I'm going to just talk about my group because I feel like, well, I know my group the best, but also it has a really good, diverse group of people. So I'm only going to talk about the development side of my group as opposed to the validation side. But in my development group, we kind of split into process development and analytical development. And we have 10 people right now. So of those 10 people, one's my manager. He's got his master's in biochemistry. We've got 2 PhDs, and they're both in the process side of things, but more often than not, they are more with the process stewardship. So they're doing a lot more like statistical analysis of the current process, doing CPV, continuous process verification, analyzing and stuff like that. So they're at their computers a lot. And then one other person in the process side is, he focuses on fermentation, and he has his master's. So I have just my BS. On our analytical side, everybody has their bachelor's. We've got bachelor's in mathematics, we've got environmental science, analytical science, and biology. So we've kind of got a good group of different backgrounds, which is encouraging because I don't know if I felt kind of burnt out when I was done with school and I wasn't really feeling the PhD path. So I'm happy that, I found a gig that I didn't necessarily need one.
Hi, Ashley. We have another question via the Q&A. So this is from an anonymous attendee. It asks, how much continuing education do you feel you get? Are there interesting classes or programs you have to get or take while on the job?
That is a good question. So GSK tries to have you own your own development, and what that looks like is, what is it? It's like 70% on the job training, 10% formal classes, and 20%, what's the 20%? It's something else. Sorry. But the point is they do want you to look into your own development. I haven't taken too many classes, but I did attend a webinar today about lyophilization and using what was it? I want to get this right because I learned something today. Non-destructive moisture test for the lyophilized cakes. So there's definitely opportunity to learn more about what you're interested in. And they like to provide you with those means to have that extra training.
Can you speak more about the the general nature of adjuvants, and then more specifically what you're working on, as long as it's not completely classified?
Sure. So I guess, let me see. What about adjuvants? I'll question your question.
So for students that again, just sort of rehash what an adjuvant is and what it does, or why you're making them, what the primary purpose in this case is.
Right. Okay. So yeah, adjuvants basically are an additive to vaccines that help them work better, faster, stronger, and for longer, so that you're going to have a better immune response, and that's really the main goal of them. In regards to COVID, our site doesn't produce this adjuvant, but GSK has offered to work with multiple companies to provide what we call our pandemic adjuvant system, and it was called that before this, so I guess take that as you will. But it was originally for influenza, but it's ASO3, and it's an oil and water emulsion system that has squalene and a vitamin E derivative. And so in the context of COVID, that would allow vaccine manufacturers to use less antigen, which means more doses, which means more people can get protected sooner. So I guess that's my little spiel about adjuvants. The reason why I also wanted to talk about adjuvants is because GSK has so many different sites, and they also, there's vaccines, pharma, and then consumer health, which is kind of becoming its own third thing. They merged with Pfizer, and they're trying to have their own consumer health thing. So they're really focused on the vaccines and pharma part of it. But if you are trying to get an interview or at our facility, like it's good to know that, we make vaccine adjuvants, and it's like, well, I want to, make all sorts of different vaccines, and that's great. And we technically help out with a lot of those things, but we don't, make the vaccine at our site. So that's why I wanted to nail the adjuvant part of the talk. So.
I have a follow-up question in regards to adjuvants, and I was curious if you know how do they begin developing new adjuvants? Like, is there some sort of like preliminary testing that they do to kind of, start different adjuvant development?
That's a really good question. So adjuvants have been around for a long time, but typically most vaccines have used alum. So like MPL with Dr. Reby coming up and working with MPL back in 1980, that was definitely kind of a, stepping into a new frontier of adjuvants, basically. So as far as, them making adjuvant systems and like pairing, MPL with QS21 or MPL with cholesterol or whatever the formulation looks like, I don't totally have a firm grasp on how they determine which blend to use, if that makes sense. But I'm sure there's a team dedicated over in Wavre, Belgium, that figure all that good stuff out.
We're starting to get a few more questions coming in, but I did get one via email. And a student asks that the student initially went into the degree hoping to create new pharmaceutical products. However, stories like Purdue with OxyContin or AbbVie and their aggressive patent protection of Humira have pushed this particular student away from work in the industry. Can you give some positive insight about pharma?
Yeah, you bet. So I can definitely understand the skepticism and, general negative feel when you hear stories about that type of, doesn't make you feel good when you hear those types of things, right? I guess I'll just talk about in the context of what I know, and that's the vaccine part of it. And vaccines are they're a hotly contested thing, which is crazy to think about, but a vaccine hesitancy is one of the leading issues globally. And it's, I don't know, I think about, how COVID isn't something that the world was necessarily thinking about or prepared for. And you think about if, this is basically the world without vaccines, and it's not great, right? So I guess thinking about it in the context of that, I feel good going into work every day knowing that something I'm doing is eventually going to reach a patient and help them. I think one really good example of at least the application of adjuvants is that our AS01 formulation has been used in Mosquirix, which is it's in phase 3 vaccine against malaria. So that's still in the works, and malaria is such a tough thing to tackle because it's, a parasite rather than, a virus or anything like that. So the vaccine is so far showing positive results. I went to a talk here in Hamilton, I don't know, maybe 2 years ago, and one of the issues with malaria is that when you're what did the guy say? He said, like, when you're a child and you like, that's your best chance at getting a vaccine for it, so that the way it works is like your immune system. I wish I could say this better. I'm sorry. The point is, I like the fact that, you know, my company is trying to do something on a global health perspective rather than just like looking for the next buck or, you know, stuff like that. So that was a very long-winded way, and I don't know if I quite answered your question, but yeah.
So we have another question here in the chat from Reed. He asks, what is your opinion on working at an established company, GSK, versus the Blue Marble startup company you mentioned? Was there a balance between the ability to create and explore more at the startup, or did it feel unorganized?
That is a really good question. So I only had the internship there, but I was actually going to, I mean, it was pretty much gonna work there post graduation, and about 2 days after walking for graduation, I found out it wasn't gonna work out. So as far as it being organized, I don't know, that kind of speaks for itself, and they're actually not around anymore. Which that's kind of the high stakes game of being in a startup. But I enjoyed my time there. Like, it was definitely interesting. I worked on a project that looked at extracting carboxylic acids, either using a physical method or a chemical method from waste. So their whole goal was to look at, you know, waste like coffee grounds or tomato like pomace and all this stuff that, you know, gets generated from people or industries, and try to extract stuff out of it to basically create drop-in replacements for petroleum products. So, you know, it was definitely, you know, noble cause. It was really cool. It was interesting. I wouldn't say it was necessarily freer than my current job as far as you know, ability to explore. But again, I was only there for about 3 or 4 months, and you know, part of it was like, okay, I got to order my supplies and then wait for it to come in. So I didn't get a whole lot of time to get my hands dirty in that job.
I'm going to follow up on that with Gretchen's question, which was, how did the three different types of internships affect your choice of career? And I'll add my own caveat on that. Can you also talk about having to adjust on the fly? Because as you said, you thought you had a job and then all of a sudden you didn't. I think this is relevant for students that are going to be graduating soon. What options did you explore when that fell through, and how did you get, you know, come through? So again, just how did the 3 internships affect your career, and then, you know, talk about what happened when that fell through and how you got back up on your feet.
Yeah, sounds good. So the 3 different internships were all, you know, kind of more or less the same type of work, in that I was, you know, at the bench working, and that's what I really like to do. I like being in the lab. I like writing about what I did in the lab. You know, it's fun for me. I like looking at new technologies. I like learning about different types of equipment. So I think they all kind of culminated into this job that I have now, where I get to do those things. So while it was different in the sense was one government, one academic, and one startup, they all kind of still had the same theme. So it kind of just reaffirmed my interest in what I like to do, which is mostly lab work. So, and then as far as the whole internship thing not working out as a full-time job, I applied for a lot of jobs, and that gets to be kind of a downer after a while. And I guess as far as pushing through, you know, I guess now that I'm on the other side of it, when you're applying and you're not hearing back for, you know, month, 2 months, 3 months goes by, you're like, okay, well, did I just like not get past the computer generated screen, or, you know, are they still looking? You know, what's the deal? We have a job open in my group right now, actually, and the goal, the dream, is to have them started, you know, soon, like in November. But that's been open for about a month. My manager has received 3 waves of resumes, which come from an external recruiter. So your resume goes to this external recruiter who eventually weeds through the things and then gives it to, like in this instance, my manager. And now my manager is on vacation for 2 weeks, so we haven't even talked about candidates. So like, that's kind of the unfortunate reality, at least in this very specific instance, that, you know, there's a lot going on sort of behind the scenes that you might not necessarily know about. So while you're kind of there waiting to hear back from anybody, it's not necessarily a reflection on you, is something I would like to say. And as far as, I guess, my part of the story of, you know, what did I do, is I just applied for a bunch of jobs and eventually kind of got lucky in the sense that there was an opening, contract opening, and they were looking to get someone on right away. Especially with contract workers, you know, they're not necessarily looking for, you know, someone to stick around, because usually legally they can't be more than 18 months, or it's like a year to 18 months. So they're like looking to get it filled, get them in, get them started. So that's kind of what ended up working out for me.
Excellent. I have another question kind of in the similar realm of topics from Eden, and they asked, how has COVID affected your work and ability to collaborate with coworkers?
That's a really good question. So back in, well, so March is when everything got more serious in the US, right? So back in March, they encouraged us to work from home if it was possible. Since my job is, you know, about half time in the lab, half time writing up stuff, I've still had to go on site, but GSK as a whole company requires masks, which is really nice. And I feel more confident going to work, especially since there's people who don't necessarily believe in wearing masks. So it's nice to go to a workplace where they enforce it. It's mandatory to wear masks. So like from the safety point of view, it feels comfortable going into work, knowing that everyone's kind of more or less on the same page and going to do the best with it as they can for the fellow coworker. As far as collaboration, I've definitely been working home more often, and we use Microsoft Teams and stuff like that. And in some ways, it's kind of, you know, it's nice. We have, my group has a stand-up meeting every Monday, Wednesday, Friday, which previously was we meet at this board at 8:30 in the morning and we talk about what's going on in our group. So now we just do that online. And at least for me, like if I don't have to, like if I want to, I can be at home, do the meeting, and then, you know, maybe look at some emails and then go in. So it's definitely more relaxed, I guess, in that sense, as far as flexibility of either working online or going in. But I feel like collaboration-wise, we've definitely been using more online tools like you'd expect, like this. So.
So we have a question here from a student, Gunnar, who asks if the kidneys of rhesus monkeys are still being used to mass produce antigens and dead viral bodies for vaccines.
That is a good question that I do not have an answer to. I'm sorry.
Austin, you can ask the next one then.
So we have another anonymous question. Do you have any advice for networking at or before your first job out of college? How can we keep preparing for the future and not stay in a stagnant position?
That is a good question. And I don't know if I'm the best person to answer it because unfortunately for all of you going through this now, it's quite a bit different, right? I'm sure it's very challenging and it's probably not going to be easy doing networking. I know your guys' career fair has probably been impacted and stuff like that. I I don't know. I mean, I'm always willing to hear from people. So like if you want to connect to me with LinkedIn or something like that, then I say by all means go for it. You never know, like shoot your shot. You don't know who you're gonna, meet. So I don't have great advice on that one.
I have a question. I'm curious, what was the most difficult part for you transitioning from college into like a full-time job?
I don't know. In a lot of ways, it was quite a bit easier just because there wasn't like homework to think about anymore. Like I go to work, I do my time there. I have a pretty good work-life balance. I don't come home and like continue to have to work or anything like that. I work on homework and stuff like that. So I don't know if there was any big challenge. I think one thing to think about if you're interested in being a scientist or a process development scientist, one of the biggest like kind of pieces of advice I have is just like writing a really good lab notebook. And I think that has probably been one of the biggest things that I do every day is, a good scientist is someone who can write down what they did and make it make sense that someone else can read it and do it, and that it's legible and contemporaneous and it's attributable to you, and you can look back at what you did 5 years ago and be like, yep, that's what I did. So I think good note taking, and then like when you're in your unit ops class, there was quite a bit of lab reports that you have to do, and I think that's a really good example of what was a really big takeaway from undergrad to my current job.
Hey,
So, yeah.
I've got a scientific question for you.
Okay.
Well, we'll see if you're familiar with it or not. So when you showed your tree bark, that reminded me of the Taxol story. Are you familiar with Taxol?
I'm not.
So Taxol it's an anti-cancer drug, and they originally extracted it from the Pacific yew tree. And so I think the Pacific yew tree is threatened or something like that. So they were cutting down trees to get the bark off to pull out the Taxol. Well, what they realized with the help of some MSU scientists, this is, I don't know if it was late 80s, early 90s, is the compound of interest that was in the bark was actually being made by a fungal endophyte. So a fungi that lives inside of the cells of the bark. So instead of having to cut the trees down, what they could do is isolate the fungi and then grow the fungi in a bioreactor and get their molecule of interest.
Yeah.
So, you doing your extractions, the thought of, you were talking about variability and things like that. If the molecule is being made by these fungi, you could imagine, with conditions, the fungi would change its behavior, maybe change its production. So the fungi is part of the microbiome of the tree. Mm-hmm. So maybe something to think about.
Yeah, no, that's actually a really good point. So to kind of expand on that, we have quite a few projects that are ongoing to either look at different options for instead of using the tree bark, we're looking at a plant cell culture, which is very similar to what you're describing. We're looking at just expanding the characterization so that we don't have to be so strict with our saponin content that we're looking for in our starting material. And we're also looking at like clonal biomass. So what that would look like was we find the tree that has a pretty good profile and basically clone it. And then instead of having to wait for a tree to grow up all the way and use the bark, you can basically make a plantation of the same clone and more or less mow it down, and you wouldn't really have to do the screening activity anymore. So there's definitely things underway. Pharma is something that takes a really long time to get anything done. So those are being investigated, but yeah, for now, unfortunately, we're having to do the screening, and that bark is, it's interesting that bark is actually used in quite a number of fields, including food stuff. So like that foaminess nature of it, it gets used in beverages and stuff like that. So other industries consume the same type of bark as well. But
And this bark is shipped from South America?
yeah, Chile.
Okay.
We have another question in the Q&A here. It asks, what advice would you give students going into an interview with a company with GSK or similar to?
GSK loves that STAR model, the situation, task, action, result. So, you know, all that good advice you're getting from the MSU like resource career thing definitely is applicable towards that. So I would say definitely get your little stars figured out before the interview.
All right, I have another question from the chat from Delaney, and they ask, how do you think your grades impacted getting a job out of college? I'm concerned that I'm going to be overlooked a lot because of my grades are only mediocre.
That's a good question. It's hard to say, because I think part of the reason why I kind of got the contract was just because I was nearby and had an okay background. And as far as, okay, she can do stuff in the lab and she's nearby. Great. So I don't know how important my GPA was in that context. I know there's advice online that, you know, if you don't have a certain GPA, just like don't put it on your resume. I think that's fine advice. I don't know. It's hard for me to say one way or the other, I guess, since I'm not really in a hiring role, but we're getting all these resumes in and I haven't looked at GPA once, if that makes you feel better.
We're closing up here. We're fast approaching 7 p.m. and we did get a little slow start, so we have a couple extra minutes if need be. So students, if you have any other questions you'd like to ask, I believe I had another one come into my email earlier today, so I just want to check on that in the interim. I sent out a link to register for the event for AICHE. It's a 3-question survey, students, so please do that. One question, what are your thoughts on continuous manufacturing and inline monitoring and sampling? So general, you know, first thoughts regarding continuous manufacturing coupled with inline monitoring and sampling. This came via email today, a question.
Well, I guess I'll take a stab at it. We have to take samples from the beginning of our MPL process, which starts with fermentation and ends with lyophilization. So we're taking samples every step of the way, depending on, you know, each step. We have the fermentation. We need to make sure the cells are reaching the appropriate optical density. Hydrolysis, we got to make sure that it's actually detoxified and ready for patient use. We look at the things that we call congeners, that makes up basically the composition of MPL. There's different congeners. So there's definitely things we have to track throughout the whole process. And then at the end of it, when it's the freeze dry powder, it goes through a whole host of tests before it can be released. And that's all focused on patient safety and, you know, product quality.
Excellent. Well, on behalf of all of our AICHE officers, Madison, our president, Austin, who's our tech support and was the person who figured out how to allow you to share your screen today, And all the students, we can't thank you enough. This is a huge contribution to our students' education. It really, as Reed, a student, says, is very informative, and thank you. These things are so incredibly valuable, and thank you so very much for taking time out of your evening to share your stories with the students.
Yeah, you bet. And I guess I'm gonna just throw this out here. I like looking at resumes, so if you're like wanting another person to have eyes on your resume, you can find me on LinkedIn, and I'll probably take a look if I have time. So yeah, I like doing it. So yeah, cool. Thanks for having me.
Thank you, everybody.